안녕하십니까? 쉬는 시간에 커피 좀 드시고 좀 휴식을 취하셨나요? 이제 저희가 크게 11개 주제 5개 세션이 있는데 3개 세션이 끝나고 이제 네 번째, 다섯 번째 세션만 남아 있는데 그 네 번째, 다섯 번째는 다 신규 과제들입니다. 주제들입니다. 그래서 이번 네 번째 세션에는 세 분의 연자가 준비되어 있고요. 첫 번째로 그 모리아 비빅 쿠마르 박사님이신데 강남 성심병원 강남중개의학연구소의 시니어 리서처로 계시고요. 오늘 발표해 주실 내용은 Development of Cell-Free Regenerative Medicine, 그래서 파라다임 시프트에 대해서 강의해 주시겠습니다. 그 비빅 박사님은 그 인도에서 오셨고요. 인도 DDU 고락프 유니버시티에서 학사, 석사, 박사를 하셨고 2011년도에서부터 17년도까지는 인하대학에서 어시스턴트 프로페서로 계셨고 인도에 가셨다가 작년서부터 저희 강남중개의학연구소의 시니어 리서처로 계시고 저하고도 같이 연구를 같이 하고 있습니다. 그래서 주로 이제 재생의학에 대해서 이제 연구를 주로 하고 계신데 오늘 그거에 대해서 셀프리 리제너레이티브 메디신에 대해서 강의를 해주시겠습니다. Dr. Bibi, please start your presentation.
안녕하세요. Thank you, Professor, so much for introduction. And actually, we wanna tell a story about regenerative medicine. That is the research going on in our laboratory. And here we will present, some you can say informatics-based analysis, or you can say study, because still we are in a process to get some breakthrough, and hopefully next year we will have some better breakthrough related to cell-free regenerative medicine from our lab. So this is outline for the today's presentation, and I just want to share a very important information why it is very important to develop, cell-free, you can say regenerative medicine. While looking, you can say regenerative medicine market, it's projected that until 2026, with the you can say annual growth rate of the 26%, and it will reach around you can say 23,800 million. It started from 2018. And maybe in 2026 it will be around you can say that is the 150,000. So that's the too big, you can say, amount. Market is going to be captured, and that's why most of the medical facilities as well as hospitals, they are trying to develop a special self using the personalized and customized medicine, using especially for the cell-free regenerative medicine. And here you can say some very important examples, that is the clinical trials which is going on related to lymphoma, heart failure, osteoarthritis. You may see that there are so many, you can say new regenerative procedures are there. While in case of the these most of the procedures belongs to the cellular system, and we know very well that the cellular system have a lot of drawbacks, especially one of the most important factor, that is you can say immunogenic rejection. And not only rejection, for example, the CAR T cell have a lot of potential, but we know that the CAR T cell have a huge side effect, especially you can say neurotoxicity and other toxicity, which is reported very remarkably during the clinical trial. So it's very important that Korea is still, one of the leader in the global scenario, while especially regenerative medicine. And related to stem cell based therapy, we have so many clinical trials as well as product in Korea itself. But in case of the cell-free system, we are still, trying to become the leader in the world, and that's why the idea behind entire story. And if you just go with the scientometric analysis, what kind of the publication trend is going on, so you can just see that from November 2021 to this last November, there are about 18,000, more than 18,000 publications related to regenerative medicine, and out of them, that only cell-free regenerative medicine, they are talking about 2,000. more than 2,000. It means there is a big gap. So we can focus over there. Our lab is focusing to you can say fill this particular gap, and that's why our major focus for the regenerative medicine development from the cell-free system. So this is the overview strategy for the, you can say, developing regenerative medicine. It can be divided in mainly three parts. One is cellular therapy, which is very old and classical, and still it is in practice. The another one is your material therapy, that a lot of people are using this kind of the material therapy. They are injecting certain kind of the biopolymers and etc. While the molecular therapy is controversial as well as very important also, because we know that it has a lot of potential, especially in case of the molecular therapy. It comes with the combination using biomolecule along with the genome editing, and CRISPR provide a very important, you can say, breakthrough, especially related to the cystic fibrosis. So that's the very important breakthrough had been made. Now, in case of the, as we know that whenever we try to inject any new biologics into the patient, so obviously the immune response is very critical, you can say, factor while deciding any kind of the regenerative medicine. And we know that till date, the you can say autografting is only possible, while we go with the allografting, because unless we will go for the any optimized allografting, the cost will be not, you can say, minimized. And if the cost is not minimized, then everyone cannot afford this kind of the therapeutics. And cell-free allow, you can say, to minimize this immune related rejections. So we know very well that in case of the host immune response, because you all know about that, there are so many things, I mean, about you can say foreign body reaction. And in case of the foreign body reaction, it recognized as a foreign, so obviously there will be, you can say, antibodies developed, and that antibody go in memory cell, and then T cell mediated, you can say, squeezing is there. It's a normal procedure. And whenever we are injecting any type of this cell which have any kind of the unique marker, then definitely it recognized as a foreign, and then antibody is developed. And instead of the treating the patient, it again harm the patient. I mean, the harm is very higher than the treatment effect itself. So here it is very important that how we can transfer our knowledge to the clinical, you can say, application. And thanks to informatics, I mean, nowadays we are not going, very lengthy process of the clinical trial. So many things we can perform, I mean, by in silico method, then we go with the preclinical data evaluation, and after that we can go with the clinical trial. But still, I mean, our process is very much similar to, you can say, the previous studies say that whatever information we have, and you can say translating into clinical application, there is a big gap. And you can say cell-based therapy is very, very powerful, but there are so many, problems which I will discuss here. I mean, you can see that what is cell-based therapy. So still in our scenario, the cell-based therapy is basically, you can say, related with the mesenchymal stem cell. The mesenchymal stem cell have a lot of differential potential, and we believe that this kind of the stem cell will, you can say, heal the body itself and can achieve the homeostasis of the body. So obviously, I mean, people are trying to inject the mesenchymal stem cell, I mean, from different origin, like an adipose-derived mesenchymal stem cell is nowadays very popular, because we can get a good amount of, I mean, good population of the stem cell from there. And stem cell story is you can say stem cell therapy story is not new one, because we already know that bone marrow transplantation is basically a kind of the stem cell therapy itself. We are taking the bone marrow and injecting the patient, and then patient will revive from there. So stem cell have potential to, you can say, heal most of the, you can say, body part, but again the problem is that their survival rate, their success rate, and most important, acceptance rate. So challenges is very important related to cell-based therapy. Number one, that is the cell, you can say, extracellular matrix interaction is very important, because whenever degeneration is started, then matrix behavior is very much changed. So whatever cell we are injecting, that is not going to adhere with the matrix, as well as there are you can say minimum vascularization is there, or nutrient deficiency is there. So that is one of the biggest factor for survival of stem cell into the you can say when we are injecting into patient. The second one, mechanical stress. We know that isolation, production, and injection have so many mechanical work. Like first we have to grow the cell into laboratory. Then we are harvesting by centrifugation method. So there are so many damages there. Then we have to you can say check. Injecting by the syringe, and it requires so many mechanical sear and tear type of the pressure. So obviously the optimized number of the cell insertion is usually not possible. The second one is that hypoxia and nutritional stress. As I already said, that most of the time the degenerated extracellular matrix, they have failed to provide the proper nutrient as well as you can say the oxygen because of a vascularized condition. And in case of our study like orthopedics, we are mainly focusing over the, you can say, tendon, and tendon is again a very tough type of the muscle where the vascularization is very least. So that's why regeneration in tendon is very difficult. And in case of the immune response, I already speak about that. The immune response is major, hurdle. Then manufacturing process, we need every hospital should have a specific kind of the laboratory, and obviously reproducibility, that they manufacture clinically effective stem cell. And we know that these are the cells. So if we try to isolate from the tissue, so it totally depends on the patient also, because how many resident cells are there and their quantification, it takes time. And ultimately, if we try to isolate from cells, so removing you can say most of the differentiating cell from the stem cell, it takes many time, more than 2 or 3 months. So obviously this time duration and technical support and payment for that kind of the work is very expensive over there. And the cost is obviously very important, because nowadays every government is willing to provide all kind of the treatment to the last man. So the cost must be an important factor to reach any new medicine to the last man. Now come to the cell-free regenerative medicine. Obviously, cell-free regenerative medicine is somewhere between, molecule and you can say cell. It's because it's mostly the cell-free regenerative medicine are secretome, whatever the cell is secreting. So that secretome have growth factors that have regulators like cytokines, as well as it have exosome. So I must say that exosome is just like a powerhouse. So this powerhouse have so many type of the factors, which includes epigenetic regulators like miRNA, siRNA, non-coding RNAs are there. And also it have some mRNAs. And this particular exosome also contains some receptors, so they have ability to target their, you can say, specific delivery site. So this is again a very important factor. So whenever we talk about the exosome, we know that we are using like a parakines type of the cell-free system. We usually classify it as a growth factors. And the effect of the growth factors are very much, very well recorded. And here there are some, you can say, still used growth factors like VEGF and FEGF, TGF beta are there. IGF we are using. Then extracellular vesicles, these are right now, the entire story about the 10-year when it get the boom, because most of the researchers found this particular exosome have lot of potential because it includes surface marker, surface receptor, as well as it have so many biomolecules which can reprogram the cellular system. And if we are able to reprogram any cellular, or you can say cellular system, so obviously the tissue can be also bring at homeostasis condition, and it have potential to treat cancer, as well as it have potential to proliferate the cell to achieve the normal, you can say, healthy tissue itself. Then polymer scaffold. So obviously I belongs to orthopedic department, and the story is not hidden that we are using so many scaffold, as well as you can say acellularized matrices for application of the orthopedic surgery and etc. And the gels is still in practice. However, the gel maybe, in future we will have a gel can be like immobilizing material. Obviously, this will be provide new insights for related to development of cell-free regenerative medicine. I already speak about that, you can say cytokines and growth factors. How earlier it was believed that cytokine is different and growth factor is different, but now it collectively we read that cytokine and growth factor, they work together, and the growth factors are signaling molecule that regulate the cell proliferation, differentiation, and migration. While the cytokine was the first used to define, you can say, subset for the growth factors influencing immune and hemopoietic cells. However, it is now clear that cytokines can act on many different cells type and their, you can say, diverse effect on proliferation, differentiation. Therefore, cytokine is now used more broadly as interchangeably with the growth factors. So here these growth factors has been properly documented with you can say, and their role, like angiogenesis, you can say VEGF and FGF, these growth factors, and also TNF. We know that it increase the circulatory, increase circulation of monocyte and monocyte induced. Different type of the functions are there. So as information is growing, but very important thing that in our body, we don't know what is the optimized concentration. Every patient, every individual respond in a different concentration. So obviously we have to, you can say, think about that, how we can use this growth factors as a regenerative medicine. And there are so many, and still we are using in our normal practice. So doctors have to decide that, okay, this patient condition is X, so can we provide certain amount of the growth factors for proper healing or not? And truly it's, and the response is patient to patient, variable. So that's why we don't have any optimized, you can say, protocol related to growth factors. While exosome was very new, you can say, baby of the biology, advanced biology, because in 1983 it was first, discovered. But however, most of the research on exosome has been done in last 10 years. And exosome is extracellular vesicle, but this vesicle is basically, you can say, endosomal complex required for transport. So this is very important. ESCRT dependent as well as independent pathways are there. So if you see the exosome is very interesting kind of the vesicle that contain double, that is the lipid layer, that phospholipid layer, double phospholipid layer, and obviously it has so many surface proteins. So broadly we can say the exosome contain nucleic acid, that double stranded DNA as well as single stranded DNA, mRNA, miRNA, etc. Then the structural and regulatory protein is also there, that some have been already recorded from the exosome. And then surface protein, that is very important for addition as well as, you can say, targeting to the specific you can say signaling and etc. Then immune related protein is also there. For example, MSC class 1 and MSC class 2 as well as CD86 is there. So exosome, because it's a part of the cell, so obviously it also contains so many things. And another thing is that, as I already say, that it also has the chaperone, which can also help to guide the protein proper folding. So it has so many potential, but only thing is that optimization. So here it's a biological, origin of the exosome, and its production is very well documented, and you may see that how it is, behaving like that. So it's a novel way, as we know that the Golgi body, it gives some kind of the vesicle, and this vesicle can be, you can say, converted in different type of the you can say, functional vesicle. Among that, one is exosome. So exosome is having this kind of the phenomena, which is usually provide a specific role in a micro environment of the tissue, where it gives, kind of the cell to cell signaling and information, and it regulate that how to, cell behave, whether it has to be differentiate or still it has to be proliferative, and etc. So, and exosome isolation is very important. We can get exosome from anywhere, even from the plant also. So many people are trying to isolate exosome from plant and just want to see that their therapeutical application also. But in our body, we can get from the CSF, saliva, milk, blood, urine, etc. And urine protocol is already established. Then exosome isolation is not very big task, because the already known protocol for the virus isolation, similar protocol we have, that exosome isolation, we go with the ultracentrifugation. And also right now there are so many commercial kits available. However, these kits are very expensive. but the ultra-centrifugation system for the research purpose is very good. And we have the exosome, then we go with the exosome characterization using immunoblotting and electron microscopy, or you can say some other technique. And however, the most powerful technique is immunoassay and flow cytometry, and another one is your electron microscopy. So here, the exosome can be characterized by, I mean, you just see that their origin, like MSC derived, like mesenchymal stem cell derived, they have the different marker. If you see the matured and immature, you can say electron derived exosome, so it have some different marker. If you see that NKC derived exosome, it have a unique marker, that's CD56. Then it's a different type of you can say origin, and obviously it also contain a characteristic miRNA that is different because it has been isolated from the different type of you can say sources. And because it contains different type of the mRNA and their marker, and it have different pathway as well as the cargo type of the pathway involvement, and their role is very much you can say interesting. Like one is behaving like immunosuppressive, another one is you can say stimulatory. So we know that, I mean, if we have to optimize the condition to isolate the exosome in appropriate, you can say condition from the appropriate condition, so these exosomes can provide a better therapeutic application for the clinical purpose itself. Yes, there are so many publications suggest that exosome as an anti-cancer, and here there are this particular exosome mediated cell to cell you can say crosstalk in a you can say tumor. So you just see that the tumor, if you see the tumor, I mean, obviously there are cancer cell, and if we you can say if we stop that is the apoptosis, then obviously the cell become cancerous. It's a normal phenomena. So only balancing is very important criteria over there. So if we are able to reprogram the cancer cell to behave like a normal, then the treatment is very much you can say obvious and possible. And exosomes have one ability: they don't need any medium to be transported, because they can cross through any barrier. Even they can cross the blood-brain barrier also. So that's these you can say exosome. They have the different type of the activities are like the tumor can be like normal epithelial cell to be converted into tumor or something like that. So there are involvement of specific microRNA that has been already identified, and we know that if we regulate this miRNA concentration, then we can also control the cancer propagation, as well as you can say their control in a in situ position. So and here you can see that how this signal transduction pathway of exosome in tumor microenvironment they can control over there. So for example, I mean, you just see that there is a blood stream over there, and here the B cell and tumor cell and endothelial cell, cancer cell, so many cells are involved in one environment, and obviously they are talking together, and we need to identify what kind of the communication they are establishing. And this particular communication is established through the exosome, because exosome is carrying all the information and delivering to the cell. So we can decode that what kind of the behavior is going on, like mitochondrial phosphorylation, or you can say the post-translational regulation, as well as you can say increase the trait cell activity or inhibit the cytotoxicity. So there are so many things are there, and obviously like from M1 to M2 conversion in case of the macrophages is also regulated by the exosome. So here in this case, so precisely you can say targeting tumor with the engineered exosome as a delivery carrier is also possible, because exosome have a space, it's just like a vessel. So we can insert, or you can say a specific type of the drug, or even high dose of the toxic drug. And because exosome also have, you can say, targeted that receptor and ligand type of the interaction, so it can target to the cell itself. So we can load the drug and directly deliver to the that tumor itself. And in case of the solid tumor, the diffusion of the exosome is very very easy than the providing any different mode of the drug delivery to the cancer itself. So this is one way that we can engineer this particular exosome for the specific type of the cancer, and then drug can directly go to the cancer, and obviously cell will be moved towards apoptosis, and we can control the malignancy. And our laboratory is mainly focusing that another side of the exosome, because exosome can kill cancer, and we think that exosome can also promote the cell growth, especially in degenerated tissue. So we are looking in this direction, that exosome in the wound healing and tissue repair and regeneration. So here the exosome also contains so many miRNA, which has been reported especially for the regulation in and related to their specific growth factors is included in this exosome. And we can find out that whether this exosome can be used for the wound healing or not. And lot of research say that adipose derived exosome as well as you can say some other mesenchymal cell derived exosome have lot of healing potential, and people have already validated their data by preclinical you can say study. And this gives new insight. And if you see that how this you can say major regulatory pathways and factors has been affected, you especially related to your exosome. So in this case, you just see that the major regulatory pathway released by the you can say exosome as well as growth factor, you know that like VNT pathway or IGF pathway or eutrophin pathway or TGF-beta's pathway, or you can say VEGF pathways, are very important for wound healing or regeneration process. You can say cell reprogramming is very important. So whatever the resident cells are there, if we want to convert them into highly proliferative states, then we need to provide the medicine or therapy which can properly regulate these pathways. And exosome already documented that their involvement in these pathways and their you can say positive role in a regulation of this kind of the pathways for the proper cell reprogramming. Now same you can see that like we know that the expression of the KIMP1 that high level in patient with non-traumatic rotor cuff tendinopathy, and it can influence the matrix metalloprotease activity and determine the normal healing remodeling. So even if we have the high number of the cell, and after that if suppose it has been differentiated but the remodeling of the tendon is not there, then again it forms the scar instead of the normal tendon. So we try to heal. There are 3 steps. One is cell division stage, number 2 is cell differentiation stage, and number 3, the matrix remodeling should be there. So remodeling means they have to attain their previous structure. So in that case, the matrix metalloprotease play very important and vital role, and we want to regulate this remodeling step itself to achieve the healing and avoid the you can say further tear or shear type of the rupturing and etc. So here you can see that what is entirely there that's working. So role and mechanism of the adipose derived stem cells, exos in treatment of the tendon injury. So our entire research is related to adipose derived you can say tendon, adipose derived stem cell you can say exosome. And here they promote the angiogenesis. They also you can say decrease the inflammatory responses, and they induce the cells, resident cells, to become proliferative more and make more tenocyte. And finally that the synthesis of the collagen as well as you can say remodeling steps are there. So we are closely working that which type of the exosomes are very you can say and which type of the function are correlated to each other, and then further we can optimize that exosome A injection. Then after certain period we will provide the exosome B to get the proliferative, differentiative and remodeling. So you can see that this pictorial diagram, it says that how it behave. After rupture, that first the cells has to go to the proliferative step, and then you can say it should be like a cell should be migrate, and it should go proliferative to make the make the tenocyte, and this tenocyte to synthesize proper amount of the collagen, and this collagen should be arranged properly, then only that will behave like a appropriate muscle, otherwise it is just like a mass. So exosome have a lot of potential. Yes, I mean we are confined only related to orthopedics, you can say application, or application for the musculoskeletal uses. But you may see that these all have been already documented, that how it can promote and proliferate the cardiac cell, cardiac muscle, I mean different type of the roles already established. And this is, you can say, tiniest, I must say that this is the miracle molecule, miracle vesicle, because it has so many type of the regulatory affairs. And if we go with the, you can say, exosome and musculoskeletal, so yes, there are so many studies are still either at the clinical state, level 1 or level 2, or some are at preclinical state. But most of the time, you just see that osteoarthritis, a regenerative disease, here they go with the, you can say, bone marrow exosome. The bone marrow exosome that you can say osteoarthritis has been rescued, and suppressing the inflammation cytokines, and also you can say NRL that inflammations. In case of the osteoporosis, that also gives exosome from the induced pluripotent cell using the that mesenchymal stem cell. That recovery of the critical size bone defects by improved angiogenesis and osteogenesis by the MSC derived exosome. And another thing is that, like osteoarthritis, people are doing so many experiments, while what kind of the molecule from the exosome is involved. So exosome is okay, then subset of the molecule, like whether miRNA or growth factor or any cytokines are involved in promoting this kind of the osteogenesis. And here some more studies is still going on. That's why we are working on exosome in our laboratory. But today morning, I mean, Professor No, he just told me that what about our future related to osteosarcoma? Then I say okay, and he just suggests me some articles that why not you are including in our study. Obviously in future we will also focus over there, that exosome from different sources, they have different ability. And right now people are talking about CAR-T, and it has been observed that that the therapeutic potential of the CAR-T cell derived exosome, and people are looking that CAR-T cell derived exosome have their they have a tremendous, you can say, therapeutic application. And only thing is that we need to optimize and how we are isolating the exosome. So that is very important. So this particular you can say study is basically a review study, but they compile so many study, and they say that yes, if exosome can regulate the microenvironment, so once we are providing the CAR-T cell, we can minimize the using the exosome remodeling, as well as you can say mediated reprogramming, to minimize the toxicity effect caused by the CAR-T cell itself. So, and in this case, Nature Communication, you just see that the CAR exosome derived from the effector CAR-T cells have potent anti-tumor effect and low toxicity. So yes, I mean, if we got the molecule which can inhibit the cancer, but it doesn't provide any kind of the side effect, so that tool will be much better. And yes, I mean, it's a matter of the research. Maybe after 5 or 10 years, we will have more insight and more knowledge about that, and we can focus over there. However, the research is very, you can say, concrete evidence about the exosome in drug delivery. So if you see the exosome in drug delivery, that is that people are already using, because we can engineer their surface, so we can add drug as well as you can say target molecule. So it just go to target the cell surface and then deliver the drug. It's you can say desirable site. So here there are so many exosome people are trying to use as a drug delivery system. And the story is very simple, that we can have like engineered you can say targeting exosome for the drug delivery. This is model number 1. So we have the plasmid. That plasmid should have the recombinant, we have to insert the targeting peptide, and then it should be transfected to the exosome derived cell. And after that, we will have the exosome, which should be, you can say, that impregment with the drugs, and then it is given to the receptor cell. Obviously, receptor cell will receive this drug, and this drug will directly go and you can say perform their action which we need. Whether this is the kind of the how I can say replication inhibitor or any kind of the that the molecular killing molecule is there, so we can use to deliver this particular over there. The another approach that how we can go for that, so we just have the like membrane protein targeting you can say protein. So this targeting protein is specific binding between the protein and drug, and then it just go and bind with the drug, and after that it deliver to the cellular system, and targeting you can say drug carrying exosome modified with the targeted protein bind specifically to the target cell. So this is the second approach. People are trying to go for that. And here this is the third one, that we can take the exosome, and this exosome have the you can say ligand protein, that magnetite nanoparticle, and it should be added over there. And after, because it's a magnetized one, so we can guide properly that where it has to be go. Like for example, engineered drug carrying exosome should be given there. Then external magnetic force will allow to you can say concentrate all the exosome at one specific site, and then it will be you can say act at the peculiar site itself. So this is the another type of the approach they are doing. But yeah, I mean, you may ask, I mean, yes, whatever I am speaking, it have any, you can say, concrete evidence whether some clinical trial is going on or not. Yes, there are so many clinical trial is still going on. We know that the clinical data related to exosome for use in treat different diseases. So you see that, yes, still the mesenchymal stem cell is major source for different type of the disease like multiple organ failure, like you can say that is the severe COVID pneumonia, then different type dry eye, type 1 diabetes. In case of the dendritic cell, people are looking for the sepsis, you can say non-small lung cancer, then metastatic melanoma. Then the plant, people are also, as I already say, that some researchers, they are extracting exosome from the plant source, and they say that it can be help in that colorectal cancer, obesity, head and neck cancer, then some other kind of the diseases. And the important crop, as we know, that the ginger or grape extract, which we already know about, that curcumin is another one. And this is the phase and patient you can say involved in the study, and you can also find that what is the development related to the information from the government side. Now I'm coming to the last stage of my presentation, that why we are selecting this exosome. Because when we go with the stem cell, I already say that the manufacturing is a big challenge. Every time we have to make the new cell from the patient itself. So general culture condition is normally used, but a specific culture condition might be, you can say, needed to produce a specific cell. While in case of the secretome, it's a general culture condition can useful for the, but a special culture condition, you can say, mimicking the pathological condition of the target disease. So we can tune different, for example, if we just add certain molecule which can give a specific type of the exosome which help in a specific condition like cell differentiation or cell inhibition, or you can say matrix remodeling. So obviously we can provide or tune our entire culture condition, and this can be given to the patient itself. And in case of the quality control, we need, both required very highly sophisticated quality control, whether it's stem cell as well as you can say biologics. So the quality control is very important, and characterization is complex, but nowadays we know that a specific marker is already reported from the exosome. So flow cytometric method based method, or immunochemistry based method, we can characterize the exosome population itself. Then cost of you can say production and treatment. So cost can be reduced by a large scale expansion of the allogenic stem cell, but till date the allogenic stem cell therapy is very difficult. And in case of the secretome, we can make, as much as amount we want, because the large scale production, and then we can store this material for the further use also. So I try to make honest, comparison for person who are looking for that. But yes, while we are working over there, to most of the time we are looking for the positive side of exosome itself. So as per advantage, both have certain advantage, like stem cell can be differentiated into a specific lineage. In case of the secretome is mainly dependent on the number of the resident cell itself. If there is no, you can say, effective number of resident cells are there, then you cannot regenerate, and vice versa. But in case of the stem cell, it can give a new cell. So that's why this is the one of the biggest advantage with the stem cell using the regenerative therapy. The another thing is that the cell cannot cross any barrier, but in case of exosome, they can cross any barrier, like blood-brain barrier, they can cross easily. In case of stem cell, stem cell also have so many advantages, like can reprogrammed into pluripotent cell, and also can cross. It can cross the blood-brain barrier, and here in case of exosome, the lower risk for the obstruction, they are smaller in size compared to stem cell, easier to store, and can you can say degenerate or senescence in a host after the transplantation if it is not required. So this is, their chances for the carcinogenicity is very low. Now disadvantages, every technology, every technique came with the advantages as well as disadvantages. So as in case of the stem cell, it have high risk for the mutation and carcinogenicity, while in case of the secretome, it cannot be used as a cell replacement therapy, as I already say that it highly dependent on number of resident cells. Then ethical issues, obviously the ethical issue is a major concern related to cell-based therapy. Then you can say might induce the host cell immunity, and rejection chances is always higher. Then cell degeneration or senescence in the host, as I already say that there are so many factors which can influence the effectiveness of the cell-based therapy. Then potential vascular, you can say, obstructions is there, and the most important thing is that optimization is very important criteria. But in case of the secretome, if you see the disadvantages, yes, I already say that it depends on the number of the cell. Then lack of understanding on the mechanism of action. Then lack of long-term safety data is there. And the lack of, you can say, standardization is always there. But because this is very new technology, so obviously once the engineers come in this particular discipline, they will provide a better, you can say, standardized protocol as well as the storage capacity. Short half-life, most of the biologics have their short half-life. It's nothing to do with the stem cell or you can say secretome. The difficult to purify and specific therapeutic component. Right now there are so many commercial kits are available which can specifically purify your exosome from the fluid itself. So once the requirement will be in a mass, so after engineering we can have the cheaper production unit over for the exosome. So this is advantage and disadvantage. So yes, exosome have potential as an appropriate alternate to conventional and generic medicines. Then cell-based therapy, it can replace the cell-based therapy as well as gene therapy. Then cancer therapy, we know that nowadays there are so many types of the cancer therapy, including amputation to you can say your that chemotherapy and etc. So this can replace this particular therapy also. Then tissue replacement therapy is also possible because the exosome have potential to induce the cell or reprogram the cell to become proliferative and further give, in situ, you can say, production of cell and their differentiation and further remodeling of the extracellular matrix. So this can be achieved over there. But at this, you can say, current early stage exosome studies, we don't have, complete understanding related to, formation, etc. So we want more insight from there. But I believe that with the continuous research work, in the near future, we may make full use of the advantage of exosome as natural carrier, as well as you can say bypass the shortcoming, and great progress will be made in the therapeutic development. So, most importantly, when we say the exosome as an alternative, we must consider the biology, biochemistry, biosafety, omics data, as well as you can say engineering, economics, and ethics should be considered to develop exosome as well as you can say any secretome as a cell-free therapy. So you may also follow these good articles where so many insights over there. And thank you so much. This is our entire team, and Professor No is our director over there, and we are working in a small group, but hopefully we will have some bigger you can say breakthrough related to that. And if you have any questions, please.
Thank you, Vivek. And my question is about the current definition of the exosome.
Yep. As we know that exosome, it has been defined as an extracellular vesicle, and there are several types of extracellular vesicle. And exosome here. You can see that the exosome, it starts with the, you can say, there are so many type of the vesicles are there. Among that, the exosome which contain a specific marker, including the certain type of the biologics which can reprogram the cell, so that is characterized as exosome. However, its size and you can say characteristics is different based on the markers. It can be identified that these are the specific type of the exosome. So exosome is basically characterized by their surface molecule itself. There are so many type of extracellular vesicle, but exosome is characterized by these markers which is present over there, and they are carrier of biomolecule which have ability to reprogram the cell of surrounding cell.
Okay, thank you for your presentation. Because time limitations. Okay. 네, 다음은 그 동탄성심병원의 정형외과 조교수로 계신 김용태 교수님께서 강의를 해주실 텐데, 김용태 교수님은 한림대학교에서 트레이닝 받으시고 그다음에 서울대에서 펠로우를 하셨는데 이제 같은 정형외과긴 하고 강의를 전임이나 주니어 스태프들을 위해서 제가 부탁을 드렸는데 저희가 이제 논문 쓰다 보면 참고문헌 같은 걸 이렇게 정리하고 그러는 거였는데 그 정리나 찾거나 이런 자료를 얻거나 하는 거에 대해서 좋은